Hello everyone, my name is Laura Gould and I'm the Research Assistant Professor at NYU Grossman School of Medicine, and I am the co-principal investigator of the SUDC Registry and Research Collaborative at NYU Langone Health. Today I would like to give you a summary recap of a webinar we did earlier this year on January 23rd, 2026. This was a presentation given by myself, Dr. Davinsky, the co-PI with me on the study, and Dominique Leitner, our senior research scientist, neuroscientist. Due to a technical glitch on my end, I did not record the presentation correctly, so we could not post the video. So I thought today, after a few requests, I would re-record it with my summary notes going through our PowerPoint deck to be able to provide the information for those who missed the webinar and we're hoping to receive the information. So thank you in advance. I'll just kick off by talking a little bit about where we're located. We're located in New York City. Physically, if you're familiar with New York City, from my office window, a few avenues away is the Empire State Building. So we're kind of in Midtown in the East 30s in New York City, but you can see that NYU Langone Health is a large institution that goes across the 5 boroughs of New York City, Long Island, Connecticut, and also as far away as Florida. We have many rankings and honors at NYU Langone Health that we're very proud of, including 13 specialties nationally ranked in the top 20. Our Department of Neurology and Neurosurgery, where Dr. Davinsky and I, Dr. Leitner and I are all based, has actually been ranked #1 in the nation for the last 4 years in a row that we are very proud of. So getting into a little bit about the SUDC Registry and Research Collaborative, we created it in 2014, and our goals have been consistent. We want to understand why some children die with an unexplained cause of death. And we want to be able to identify children at risk to be able to prevent further tragedies from occurring. This is a very, very large effort going on that is growing. Our name is specific, the SUDC Registry and Research Collaborative. We set out to create a very, very large registry with the commitment and courage of families, but also to allow many different researchers to collaborate with us, of us doing the work with families to collect the data, and then to work with many, many different researchers from different specialties. So you'll see our research team at locally at NYU, the core facilities that we use at NYU. There's many others that we could if we ended up having other research projects that we could benefit from using them. And then we have many external collaborators, those at the Mayo Clinic, Children's Hospital of Philadelphia, Princeton University, as far away as Monash University in Australia. And then we have many external collaborators across the country, many pathologists of different specialties that we're really grateful for them working with us. To understand a little bit of the timeline of just the history of SUDC research, really began back with a registry that we first started in San Diego. It was called the San Diego SUDC Project with Dr. Henry Krause. That produced our first landmark publication in 2005 that defined SUDC as the sudden and unexplained death of a child 1 to 18 years that is unexplained after a thorough investigation. When Dr. Krause retired in 2011, I looked for a new home for this research and looking at what we learned in the first 10 years and what could we do better in the next, and what different approach and methods could we use to really push forward the research. And that was the culmination of creating the SUDC Registry and Research Collaborative. You'll see now in 2026, all of these bubbles off to the right, all the different areas of research that we are embedding ourselves in to uncover clues, risk factors, and the underlying causes of currently unexplained deaths. You'll also see at the bottom some SUDC research interest. This looks at research publications over time, and you see that we have been successful since the start of the registry in terms of really increasing the amount of research done in this area. If you're following on our new Instagram channel, I'll give the link at the end. I did go over a pretty in-depth explanation of this hierarchy of evidence pyramid, but really we're talking about the different types of research designs correlate to a different level of quality of information. And a lot of the SUDC research has been towards the bottom of this pyramid, but in the last really 4 or 5 years, we've been able to actually creep up in terms of being able to do higher quality designs, which gives us better quality of evidence for what is causing some of these deaths. I already mentioned this definition before, but again, SUDC is the sudden death of a child 12 to 18 years that is unexplained after a thorough investigation. A couple terms that I'm going to mention. One is febrile seizures. And febrile seizures are provoked seizures in young children. They're associated with fevers or illnesses, and they're actually the most common convulsive event in childhood. About 2 to 4% of all children between 6 months and 5 years in the US will experience one. And they're actually have higher incidences in different parts of the world, like India and Japan, where they see closer to 5 to 10% incidence among young children. They tend to occur a little bit more in boys. The peak age of onset is 18 months, and some children will have a recurrence, about a third of them. The genetic components of febrile seizures are not well understood, but it's important to note that some childhood epilepsies first present as a febrile seizure. And so that's an important connection to follow up on in terms of understanding that some children, although most of them, febrile seizures are benign, they grow out of them, there's no ill effects, sometimes it's a precursor to epilepsy. And febrile seizures can be simple or complex, as this figure shows. Simple are briefer, they only happen once in an hour, and they're generalized. They look like they are responding across their whole body, versus complex are different in some way. Either only one part of their body looks affected, they're longer in duration, or they recur within the same 24 hours. And what's important with febrile seizures also is to understand that after you have a convulsive event, there will always be a post-ictal phase. And that time is right after the convulsion completes. This is a time where the body is recovering. The person may look very drowsy, confused, a child may fall asleep. And at this time, they don't have their normal reflexes that they normally would outside of a seizure event. In contrast, epilepsy is associated with unprovoked seizures. So they don't have a fever or illness around this, although they may, but by definition, epilepsy is 2 or more unprovoked seizures or 1 unprovoked seizure with abnormalities by an EEG or an MRI scan. Epilepsy is quite common. About 3.4 million people in the US have epilepsy, and the underlying causes of epilepsy, about 2/3 of them are unknown. So they also are coming on and developing these diseases where it's not well understood why some people develop. There is an associated sudden unexpected death in epilepsy, termed SUDEP, and this is a death at any age in someone with epilepsy without injury, drowning, or other cause, and it's estimated that this occurs about 3,000 times each year in the US. In terms of risk reduction, it's shown that the control of seizures with medications or surgery helps to reduce the risk, improve a lifestyle, especially about adherence around anti-seizure meds, having a good sleep hygiene, not being sleep deprived, avoiding excess alcohol, for example. And another one is regarding sleep supervision. Recommended that someone has supervision during sleep is shown to be associated with a decreased risk in SUDEP. There's some other terminology, and this starts to get confusing. Sudden cardiac death. Sudden cardiac death is a death due to a cardiovascular or an unidentifiable cause that occurs within an hour of the onset of symptoms. And you could say, well, this could be a SUDC death, and that's absolutely correct. And that's why definitions are really important to convey to people what you are talking about and what you're not talking about, because these can get very confusing. Sudden unexplained infant death is similar to SUDC, except for the age, where it's an unexplained death of an infant less than 12 months after unexplained after investigation. And then we have SADS and SUDS, and again, it can be confusing in terms of a bit of alphabet soup. But if we think of sudden unexplained death in the young as this huge umbrella, we can see here that we have the unexplained deaths in infancy. Epilepsy or sudden cardiac death can occur at any age. Sudden unexplained death in childhood, of course, is between 1 and 18 years. And we see the majority of them occurring, about 60% occurring in toddlers, ages 1 to 4. This also overlaps with the age of febrile seizures, which is defined as 6 months to 5 years. So I just want to draw your attention to that. I also wanted to remind you that we don't want to talk about SUDC as a cause of death. It's a category of death that eludes us right now. We don't understand what the exact underlying cause is for that specific child. But we know underlying etiologies and cases that we've explained can be due to neurological issues, heart problems, metabolic disease, genetic abnormalities, infection, or other unknown. And now in 2026, even just last year, we're seeing more and more researchers talk about these different areas of study of sudden death in the young and looking at them as related in some way and overlapping. And I think it's really important because we can learn from our colleagues studying these different groups, because there may be overlapping underlying etiologies and risk factors that benefit overall. Here is a little bit of that timeline that I showed you before, and what I'd like to go into now is a little bit about the progress that we've seen in different areas of highlights, at least a few different areas of highlights that we're really focusing on in 2026 at the SUDC Registry and Research Collaborative. So the 2024 statistics just came out. It showed that 378 children were certified as unexplained sudden deaths in children 1 to 18 years. Again, there was a slight male bias. A little bit more than 50% were toddlers. We also see a difference between Black and white populations with a 2.7 times increase rate among Blacks versus whites. And although this is very rare when you look at, 1 death per 100,000 deaths in some of these age groups, when you add up those 378 children, it actually calculates to over 25,000 life years lost in just 2024. And so really an area that we need to better understand to prevent these tragedies. At the registry, for families who have enrolled and participated or read some of our papers, you know that we do a very comprehensive case review, and we are well over 300 of these reviews complete. But of the first 243 that were unexplained after our complete blinded multidisciplinary review, these are some descriptors that you can think about in terms of that group of the 243 children that were deemed unexplained after the study review. So the median age was 22.5 months. So again, most children in that 1 to 3 year old range. There was, again, a little bit of bias towards male, 60%, 40% girls. Most of the children were full term. Most of them were developing normally, and the vast majority were occurring at home. This bottom left bar chart describes a few key features looking at the child's signs or symptoms in the last 48 hours. And you'll see about half, 46%, had no signs or symptoms of any illness, but 23.4% had some cold symptoms, and just over a quarter had a history of fever in those last 48 hours. When we look at the child's life medical history, we pulled out a few things here for the chart. Starting in that middle graph, you'll see in the blue, 27.6% had a history of febrile seizures. So again, that's why I wanted to go over that definition with you earlier so you understand what those mean. In the middle there, a history of fainting, or what's sometimes called syncope, just 1.3%, very low association. And in terms of any history of breathing problems or asthma, it was a little over 7% of those unexplained deaths had these different medical histories associated with them. Off to the right here, almost all of them were sleep-related. 92% of the cases that were deemed unexplained after review were sleep-related, and all of them were unwitnessed. So that is a important factor there in terms of thinking about. We also looked at what position they were found in. About 80% of them were on their belly, and about half of them were found with their face straight down in the mattress. When we look at younger children with SUDC, say 1 to 5 year olds versus over 5 years old to 18, we see different profiles emerge. The older children of SUDC that were greater than 5 years of age were much less likely to have a history of febrile seizures, less likely to have a family history of febrile seizures, less likely to have cold symptoms or fever, less likely to be sleep-related, and a little less likely to be unwitnessed deaths or found prone. They were also more likely to be bed sharing. So again, in this instance, think of bed sharing as that sleep supervision that I mentioned earlier. So this seizure association is the most significant clinical history association in young SUDC cases. At this point, about 29% of those children enrolled in the registry have a history of seizures, and 95% are fever-related or febrile seizure-related. This association was first identified back in that first paper on SUDC back in 2005, but it was based on the first 50 children we enrolled in San Diego. And I think at that point, both Dr. Krause and I and our other co-authors thought, well, maybe this is a selection bias. Maybe it's just a coincidence in these first 50 cases. But now you'll see in this purple shade here, these all represent different papers that have published very similar associations of febrile seizure with SUDC cases. And what's important about these, while some of them are case series where we're not sure whether we're getting a different representation of the population than what is accurate, two of these, the Ireland study and the UK, are national population-based studies, and they still found similar association rates. So this seems to be a true signal with SUDC. We've also published some various different manuscripts on this topic because, as I said, it is the strongest clinical association we have right now. And so trying to better understand that is really important. Can we identify children at risk among the vast majority of children with febrile seizures? Is there a way that we could identify why those children during life, of those small number of children with febrile seizures who might be at higher risk for sudden death? Over to the right here, where it says SUDEP in epilepsy monitoring units, this was there was a study done about 2012 that looked at SUDEP in epilepsy monitoring units and identified that of the children, or of the adults, these were all adults in epilepsy monitoring units, found that if there was a sudden death in the epilepsy monitoring unit, of the 11 cases that they studied, all 11 of them occurred after a tonic-clonic full seizure event, in the minutes after it, not during the seizure, but after. So as the research study has gone on, and since we started in 2014, technology has changed, and obviously technology has changed for us in our personal lives. When you think of the evolution of cell phones and things, there's also been a similar evolution in terms of baby or infant monitors that went from just sound monitors in the 1980s and 90s to in the 2000s incorporating video, and then videos that record. We were able to obtain some of these videos, and we submitted them to an analysis, and these were videos of the child's last sleep period. We collected 7 videos. 2 of them were devices that were triggered by motion or sound, so there were time gaps that made it difficult to evaluate the what actually occurred, just because there might have been like 15-minute gaps during it. But of the 5 continuously recorded videos that we obtained, all 5 of them were identified with a convulsive event. This was the first series of SUDC to implicate seizures at the time of death, and we think this data suggests that many unexplained sleep-related deaths may be related to seizures. This particular paper, even though it was a very small series, because the data was so novel, it was this window into these sleep-related, classically unwitnessed deaths, that it reached many different news outlets, NBC Nightly News, the BBC. It went global, and we raised a lot of awareness for SUDC in the process. As a result, we had more families contact us with additional videos, and we're doing additional analyses on those this year as well. In the attempts to, again, go back to that question of, if febrile seizures are so common and they're considered so benign in the general population, but we see this signal of the association to some SUDC deaths, is there something different about those children's febrile seizures that if we knew about risk markers for them, could we identify them in life and help pediatricians identify children at greater risk? So we did this case control study of children with sudden death that had a history of febrile seizures in life, and we compared them to living children with febrile seizures. And this was an anonymous survey online where we surveyed parents under both those circumstances. Either they had a living child with a febrile seizure, or they had a child who had died with a history. And what we found in this analysis was the groups were different in a couple different possibly significant ways. The group that was associated with febrile seizures with later sudden death were younger at the age of onset of their febrile seizures versus the living children. The parents of the children with sudden death cited greater development concerns. We did not get specifics because this was an anonymous survey, so we don't know in more detail what that might allude to. And there were these 2 different significant factors in terms of what the seizures looked like in terms of being less likely to drool. We also asked a few questions about the child's sleep quality and found that there was a statistically significant difference in the amount of night wakenings of the children that later succumbed to a sudden death. And our conclusions from this were that, we identified these novel associations related to age, development, and seizures, and we suggest that the data might imply that there might be different arousal mechanisms for children with febrile seizures and sudden death. And so this is propelling us forward with doing some follow-up sleep research. Another area that we pushed forward was to obtain a grant from the American Epilepsy Society to create a library of living children having sleep-related febrile seizures. The goal was to do a case control study of looking at the SUDC videos that we collected versus living controls of febrile seizures to again see, using artificial intelligence, is there something different about the SUDC febrile seizures? Is there risk that we can identify, or are there protective factors that we can identify in the living children, both hopefully to impact future clinical care and risk identification. And this is in process as well at this point. Moving to a little bit different area of the study is looking at genetics. And so these are a couple different papers that we have published. All of our papers that are published are on our SUDCRRC.org website. So please go there if you're interested in reading more about any of the details that I present. So at this point, we have completed 400 whole exome sequencings of the 400 SUDC children enrolled in the registry. And what we find among them is about 10% of the time there were variants identified that were thought to be contributory to death by the study team. Over 90% of these were trios. So we had the child and both parents biospecimens available. And this was really, really crucial. And it's really, really crucial going forward in terms of our genetics to make sure that we're comparing the child to both parents, because it helps us identify, and it's really the only way we can know whether the findings in the child are unique to them or whether they were inherited from a parent and therefore other children at risk, in terms of possible sibling care or screening. Going forward, we're doing whole genome sequencing on all the newly enrolled. When we also look at the gene functions of those 10% of children that had variants thought to be contributory to death, we see an interesting breakdown of about half of them were associated with neurological function. A little less than half were associated with cardiac function, and others were syndromic type of problems, so they affected many different parts of the body. When we think of omics, omics is the study of identifying, describing, and quantifying the biological processes that determine form and function. That's a bit of a mouthful, but if we look at this figure across, we all are born with the DNA that we have forever. Our DNA transcribes the information when the DNA wants the body to create a function, and it communicates that through RNA, which eventually creates the protein, which creates the function. So when we're talking about whole exome sequencing, we're talking about the analysis of the genes, the sequencing of the genes that we have, and seeing, is there a mislettering in any of those nucleotide pairs in any of those genes, and is that gene associated with disease, or is it suspected to be associated with disease? But in terms of the rest of the spectrum, we haven't done a whole lot of it, especially in the middle of this figure. Dr. Leitner did do the first ever protein studies, proteomic studies in SUDC that I'll talk about in just a minute, and then I'll also explain how we're impacting some of these other areas. Because with SUDC, we really want to understand, is there any problems that are occurring anywhere along this whole process that might contribute to a problem? So going from there, I'll show in a minute a hippocampal spatial transcriptomics. That's a big fancy word of looking at the hippocampus part of the brain and looking at the RNA. And this is a new grant funded by SUDC UK. We're also analyzing significant proteins that have been identified in the past, and we're also looking at newborn metabolites with a new collaborator that we're working with at University of California in San Francisco. So in terms of proteomics, Dr. Leitner led 2 different projects so far in proteomics that has never been done before. The first one looked at brain tissue in SUDC versus explained deaths, So our controls were explained deaths that we worked with our medical examiner partners. These were crucial to have really good control comparators for this to identify whether the differences in SUDC were really specific to SUDC or not. And what we identified in that paper was that there were protein differences in both the hippocampus and the cortical regions of the brain. So the cortical regions being this top area of the brain over here, and the hippocampus being internally. I think I have a better picture of it coming up. She also did brainstem studies looking at proteomics in children with and without a history of febrile seizures, and found significant differences between the two of them. So next we're looking at what we have in progress right now is the analysis of those significant proteins. Why were they greatly increased? Why were they greatly decreased? And working backwards to figure out what the function of those proteins are, and that can help us perhaps understand the underlying causes for those. So as I mentioned, Dr. Leitner will be doing this RNA level study. We're really privileged to receive this new grant from SUDC UK to make this opportunity available to us. Another area of research includes the pathology. As you saw, we have a lot of pathologists on the study, and Dr. Leitner manages a lot of our neuropathology studies at NYU. In terms of the pathology findings in general, SUDC, it's very common that there's mild pathology identified at the time of the autopsy. It's very rare to find an autopsy report where they don't list something in their descriptions. So it's whether or not those findings are actually sufficient enough to explain the death. And if a case is considered SUDC, then those findings are mild and considered insufficient to explain. There's also been a history of subtle findings in the brain, especially in an area called the hippocampus. And that little graph off to the right is from a 2021 article where we did a blinded review of the microscopic slides of the hippocampus. We had 9 different pathologists. They reviewed 67 slides. Unbeknownst to them, some of those slides represented SUDC cases. Some of them were control cases, so explained deaths, children who unfortunately died from accidents or known medical disease. And then our SUDC group, we broke up into children with a history of febrile seizures and a history without. And what this graph on the right is telling us is those medium gray shades in the middle. It turns out that about 50% of every single group, the hippocampus was considered abnormal. So in earlier research where this was considered maybe a really, really significant finding, our understanding of this has evolved over time, because now with more control type studies, we're understanding that these findings are really common in children not associated with SUDC. When we look at the reviews and what's central to the child and autopsy findings and things that we call our intrinsic factors, so these are not findings that are considered sufficient to explain the death, but the pathologist does note them. And what we see as the most common intrinsic factors on SUDC cases is an immune system related finding. So there's either a history of clinical symptoms of mild illness, or there's findings on autopsy that the body was reacting to, there was an immune response, which they can see in some of the organs. The second most common intrinsic factor was neurological related. This history of febrile seizures, or again, these subtle neuropathology findings that I mentioned, but we know that those are also common in children and might be a variation of normal. We need to understand that and do more research, which we are doing, including some artificial intelligence studies in that light. The other thing that we have in progress is organ-specific analyses. So although we have published the first 100 case reviews with some details on the pathology, we haven't looked at it far beyond in depth outside of the brain. So we're working on that now as well with all of the data that we've collected. In terms of neuroimaging, some of the cases enrolled with us where full neuropathology was done at NYU. We also did neuroimaging, so an MRI. And with this, we are now able to go back to all of these MRIs and look at, can we see differences in the structures of the brain? So I've mentioned the hippocampus here just to point out, here is the hippocampus on one side of the brain, and here is the one on the other. And you'll actually notice this is an example where there's asymmetry. This hippocampus is a little bit flatter. It looks a little more squished, would probably be the easiest term to use for it, versus this one, which is more round. So we're now doing these volume studies. We're also doing artificial intelligence studies to understand what really is the normal architecture of the hippocampus, because it's not well understood in the literature of what the normal hippocampus looks like in children in this young age group where we're most often finding these examples. In terms of the microbiome, this has not been studied in SUDC before. We do have a pilot study that we just started in using healthy SUDC siblings between 1 and 4 years of life as comparators. So, anyone who's interested in that, that is already enrolled in the study and has a sibling, a healthy sibling in that age group who would not mind donating a stool sample, that might be helpful to us. So our case review process is very detailed. Again, it is blinded, which means that all of our reviewers don't know the opinion of the original pathologist. And this is really important because we want their independent opinion. It's not a huge community, the pathology community. Often they know each other, and so this is a way of increasing the quality of the results that we have. We are also very proud to receive a different grant from SUDC UK to help us complete the case reviews through the first 400 children enrolled in the study, and we are very proud of that. We're also working on analysis of maternal and birth factors that has not been looked at in SUDC before in depth, and we're also collaborating with another colleague, comparing it to population-based data and seeing what we can learn from the registry data as well as the population-based data, which I think will be two really rich sources to be able to see if we can identify some factors that can help us with that. We're also looking at the analysis called of discordance, when our pathologists end up disagreeing with the original pathologist. And why is that? Do they have different information? Are they using different words? Are they not doing certain tests? And trying to understand that better so we can advocate in the medical examiner community for more consistency among those certifications. You'll see up here in terms of the first 271 blinded reviews, about 81% were still unexplained when we were finished. 13% were considered explained natural, meaning it was due to a disease process within the child. 1% were considered an accident, and 4% we just didn't have enough information to complete the review and provide an opinion. We're also looking at, I mentioned before, organ-specific analyses. We're looking at analysis of all of our cardiac pathology results. And what we found so far in terms of some SUDC cases, structural or physiological changes, some explained cardiac deaths within the registry that we found most commonly due to channelopathies. Oftentimes these, historically they don't leave any evidence unless they're associated with cardiomyopathy, which is a change in a disease of the muscle, which sometimes you can see under the microscope. myocarditis, which is an infection of the heart, usually due to a virus, and then a couple other cases of sudden cardiac death that didn't fit into those above categories. Infection. Infection does occur. Children can present with very minimal clinical symptoms or have a very rapid infection process going on. When those occur on cases that we're reviewing, we do access the resources through the CDC, which can do high-level infectious investigations for us. When infectious sudden deaths have been identified in the registry, those are commonly associated with upper respiratory infections or infections, as I mentioned on the earlier slide, infection of the heart, myocarditis, so it kind of cross both things in terms of heart problems, but it is an infectious source. And then a central nervous system infection, so an infection involving the brain. Everything we do at the registry is in alignment with the 2019 recommendations that some of us on the study team were involved with and co-authors in terms of the panel on sudden unexpected death in pediatrics, and the book is entitled Unexplained Pediatric Deaths, Investigation, Certification, and Family Needs. And basically the way you can read this is, on day one, all of these deaths are considered sudden and unexpected. Where, if a child dies in a car accident, that's not really unexpected. You would expect that to happen. A child goes to sleep and doesn't wake up, that's unexpected, and an autopsy needs to be performed to further understand that. At the end of the autopsy, the scene investigation, the ancillary testing, the interviews, everything that goes on for a complete comprehensive death investigation, a death will either be considered explained, where there's persuasive evidence of disease or circumstances causing deaths, and this could be natural disease, it could be due to asphyxia, it could be due to another lethal event, an environmental type of issue, carbon monoxide intoxication as an example. The case could be undetermined, where it lacks data for assessment or there's conflicting evidence. And finally, they're unexplained. This is where the SUDC deaths would go, where the case is thoroughly investigated, but there's insufficient evidence of the causative disease or event. And in these cases, the question is asked, are there intrinsic or extrinsic factors identified? So I know I mentioned this before, but intrinsic is defined as abnormal physiologic or anatomic findings that may contribute to death but cannot explain the death or represent natural conditions of unknown significance. So an intrinsic factor here might be a history of febrile seizures. Do we know it played a role? No, we don't. In this particular case, seizures, for example, do not leave evidence at autopsy because they are an electrical event similar to a heart arrhythmia. In contrast, extrinsic factors are environmental factors that may threaten life but cannot explain the death given the overall investigative findings. The most common extrinsic factor in SUDC cases is being found in the face down position after a sleep period. Now again, if this was a 6-week-old baby, we'd worry about that really being a lethal risk factor because babies at 6 weeks don't have those developmental skills to be able to lift their head and get out, especially if they're on a soft surface. But a 1+-year-old with normal development is more than capable of rolling, sitting, standing up, walking. That shouldn't be a lethal position in an otherwise neurologically intact child. So tracking these intrinsic and extrinsic factors in SUDC has been super important and valuable in research. It allows us to identify trends. It allows us to identify commonalities in a subset of a group of children. And then that allows us to ask particular research questions about that subgroup and how can we learn more and what's the next experiment to do to answer that specific question. So in summary, the registry is doing a lot of different things. They all stem from what we learn from the specific tragedy of each individual child who dies suddenly without explanation. We're doing a lot of testing and analysis on them specifically, but what we learn from them allows us to ask more advanced research questions and provide additional research experiments that you'll see outside here in these bubbles, which I talked about a few. So I hope this review has been helpful in terms of where we are at the registry, what we're focusing on in 2026, where we're going, what needs to be done, the progress that has been made in explaining about 20% of the cases that come in. Still, the majority we don't clearly understand, but we have many leads to follow up. And compared to 5 years ago, we are so much further ahead than we were. The SUDC Registry and Research Collaborative, we have our website at SUDCRRC.org. You can find all of these publications there. If you want to support directly, all the donations are 100% tax deductible. I would also like to, again, thank all of our study team members. As I mentioned, this is a huge, valiant effort. I'd like to thank our sponsors, our past sponsors, current sponsors. This slide needs additional updates in terms of additional support amazing support that we're receiving in 2026 that we will add on for our next presentation. And final, really special thanks to the bereaved families who work with us, who have the courage and the patience to enroll in research, to allow researchers to learn from your greatest tragedy. Thank you very much. All of these successes and progress are because of you. If you'd like to get in touch with us, email, the general email is there. I will answer you, or if I don't know the answer, I'll go to one of our study team members who can. And we recently created an Instagram account listed as at SUDC_research, where we're really trying to focus on educating stakeholders about research, helping you be critical reviewers of research, helping you understand what the research progress means, and answer any questions you have. We're committed to transparency in our work, in terms of the work that still needs to be done. And again, it's the SUDC Registry and Research Collaborative, and this collaboration cannot occur without sponsors who support this work, to the study team, to the medical examiners and coroners, and the courageous families that help medical research learn from their tragedies. So thank you very much. Please reach out if you have any comments or questions. Thank you.